My name is George Ngalame Jukang. Today is the 18th of May 2020. Trying to put my name in the various forms so that hopefully it is clearly seen. I'm going to be presenting two cases from our case studies. The first is going to be dealing with the regulation of pyruvate kinase under normal condition, and the second is going to be doing the regulation of glucose uptake by cells. We're going to be comparing glucose uptake in cancer affected cells and cells under normal condition, or healthy cells as we may want to call them. It's going to be a little fast because first of all, pyruvate kinase is an enzyme that catalyzes a very last reaction in glycolysis, and this very last reaction has to do with the transfer of the phosphate group from phosphoenol pyruvate to ADP, thereby giving a production of ATP, which is energy, and pyruvate. Now, pyruvate kinase is primarily activated through the controlled or regulated allosterically. We shall look at that more deeply as we make progress. There are four types of pyruvate kinase. I'm trying to make a presentation as fast as possible to catch up with my time. There are four types of pyruvate kinase in living things, depending on the organism we're talking about. Mammalian cells have theirs. Okay, the first we talk about PKM, which itself is divided into two forms. PKM1, which is the pyruvate kinase regulated in very hard working tissues or organs, such as the heart, the brain, and the muscle tissues. We also have PKM2, which is the one pyruvate kinase isoform that is regulated in proliferation cells, such as tumor cells and cancer cells, or tumor cells and embryonic cells that need fast growth. We also have PKL, which has to do with pyruvate kinase regulated in the liver, and PKR, pyruvate kinase regulated exclusively in red blood cells. That takes us now to the form of regulation under normal condition. First, we want to state that pyruvate kinase is one of the three types of the reaction that involves pyruvate kinase is one of the three types of reaction that has a very large negative delta G, and this negative delta G makes the reaction largely irreversible. Now, the things that control or regulate pyruvate kinase, there are actually three forms of regulation, but majorly what we're looking at here is substrate regulation. And the substrate of pyruvate kinase is phosphoenol pyruvate, which already say can easily influence enzymatic activity through enhancing the reaction, letting the process to operate faster depending on the substrate availability. Also, ATP and pyruvate are seen as negative inhibitor, as well as alanine also possesses a certain negative allosteric modulation. Now, the substrate for pyruvate kinase, allosteric effector for pyruvate kinase are things like fructose 2,6 biphosphate or fructose 1,6 biphosphate for mammals. The substrate PEP and the product pyruvate kinase participate in a diversity of metabolic pathways. The regulatory properties of PK, or pyruvate kinase, as a typical allosteric protein are demonstrated through such fundamental activities in the cell metabolism. We have such physiological effectors, which include hydrogen ion, potassium ion, magnesium ion, and manganese ion that regulate this activity. Moreover, with regard to the substrate phosphoenol pyruvate, the enzyme demonstrates sigmoidal kinetics, and it is activated by a biphosphate effector, which is contingent on the organism. In this case, can be mammalian, or we can talk about protozoans. In this condition, the enzyme's activity is changed through the alteration. Is a key point. The enzyme's activity is changed through the alteration of the protein conforming, alteration of the protein conformation, which is as a consequence of the effector binding at a site other than the active site. This is what I mean by allosteric regulation. We also have covalent modification. Covalent modification is a form of indirect regulation. Here, a particular modification of the enzyme structure is achieved through chemical change, which may be in the form of oxidation, phosphorylation, or acetylation. And pyruvic kinase, basically in mammals, we're talking about phosphorylation. We also have hormonal control, where by the presence of hormones such as cyclic AMP, they have a major effect on the regulation of pyruvic kinase. Studies have shown when measured at suboptimal substrate concentration, PKL is deactivated rapidly by incubation of a human liver subnatant. Now, the next one, the reaction Y isoforms 2 is characteristically found in low activity state. As already indicated, there exist 4 types of pyruvic kinases. PKM1 and PKM2 have different enzymatic properties and are both controlled by a variety of factors at various transcriptional and post-transcriptional levels. While PKM1 is constitutively active enzyme which displays interesting activity for its substrate PMP, the enzyme activity of PKM2, on the other hand, is subject to complex allosteric regulation, which is of significance to tumor progression and growth. The expression of PKM2 is upregulated in most of the cancer cells. This indicates that PKM2 functions as a promising target for cancer treatment. PKM2 protein switches between high activity tetramer form to low activity dimer form in healthy tissues. In tumor tissues, nonetheless, PKM2 tends to exist in dimer. This PKM2 dimer possesses low catalytic activity, leading to the increased anabolic synthesis of macromolecules through the PPPP pathway. Consequently, it promotes cancer cell proliferation. Our next presentation deals with the regulation of glucose uptake by cells. Cancer is a disease that has to do with the multiplication and proliferation of cells. And because of the proliferation and multiplication of cells, there is a high need of energy. So, like we earlier said, glycolysis, the process ends with glycolysis, and from there it moves to fermentation without going through without the completion of the processes like the Krebs cycle or the oxygen transport chain. So with glycolysis, where you have fermentation and the production of ATP, the process is stopped there, and the cells use that energy. They preserve the progression of the other stages to complete respiration, and the end with glycolysis, therefore reserving energy for themselves. And these cells are regulated by what we call glucose transporters. In the liver, we have the glucose transporter GLUT2. In the muscle, glucose uptake is regulated by GLUT4. In the brain, is regulated by GLUT3, and in adipose tissues, also regulated by glucose uptake is regulated by GLUT4. Generally speaking, so their uptake of glucose is high. For cancerous cells, however, the process is different. Their uptake of glucose is higher compared to that of normal cells, and they produce lactic acid instead of metabolizing pyruvate in the tricarboxylic acid cycle. Thus, for cancer cells, there exists the Warburg effect, which is an adaptive shift. This shift results in anaerobic glycolysis, which somewhat provides an evolutionary advantage to cancer cells by enabling increasing biogenetics and biosynthesis. According to this shift, the respiration of oxygen in healthy cells is substituted by fermentation of sugar, so that while normal cells meet their energy needs by respiration of oxygen, cancer cells have their energy supply in large measure through fermentation. We already indicated all of that. Conclusively, furthermore, an increase in the repression of glucose transporters like glucose transporter GLUT and some glycolytic enzymes, including hexokinase and pyruvate kinase, lactate dehydrogenase, all play a role in the acquisition of metabolic profile. The following are the modifications involved in cancer cells: higher rate of glycolysis, uncoupling of glycolysis to TCA, lactic acid fermentation follows glycolysis, and pyruvate dehydrogenase inactivates, but lactose dehydrogenase activated in cancer cells. That brings us to the end of our 10 minutes. Thank you.